Gram staining is a differential staining technique used to classify bacteria into gram-positive and gram-negative based on cell wall properties, developed by Hans Christian Gram in 1884. For this, we need a culture plate, a inoculation loop, a spirit lamp, slides, normal saline, crystal violet, gram iodine, decolorizer, and saffronin. First step is smear preparation. We have to sterilize the inoculation loop. After that, we have to take a small loopful sterile saline solution on the slide. What types of specimen can be gram stained? Blood, urine, sputum, swabs, or bacterial cultures. With the help of sterilized inoculation loop, take a small amount of bacterial culture and mix it into the saline solution. Spread the sample thinly to form an even smear and allow the smear to air dry completely. Second step is heat fixation. Pass the slide quickly through the flame of spirit lamp 4 to 5 times with the smear side facing up. This process kills the bacteria and helps the smear adhere to the slide. Overheating can distort the shape of the cells. Now it's time to stain the smear. Step 1. Primary stain. The smear is stained with crystal violet and let it for 1 minute. Now tell me what kind of stain or dye is it? It's a para-rosanilin dye. Give some other examples like Jameson violet, methylene violet, etc. Crystal violet stain all the bacteria violet in color, irrespective of whether they are gram-positive or gram-negative. After 1 minute, gently rinse the slide with water to remove excess stain. Now gram iodine is poured over the slide for 1 minute, or sometimes it's asked for 2 minutes. So what is gram iodine? It's a dilute solution of iodine. What is its function? It's acts as mordant. The iodine binds to the crystal violet, forming a crystal violet iodine complex, which helps primary stain to bind more effectively. What happens if we skip the iodine step? The crystal violet iodine complex will not form and all the cells may appear gram-negative. After 1 or 2 minutes, rinse the slide with. Now decolorization, the most crucial step of gram staining. It may be acetone or ethyl alcohol or acetone alcohol. It removes the primary stain from gram-negative bacteria while the gram-positive bacteria retain the primary stain. By over decolorization, gram-positive bacteria may lose color, or due to under decolorization, the gram-negative bacteria do not lose the color. Why do gram-positive bacteria retain the crystal violet stain? Thick peptidoglycan layer traps crystal violet iodine complex. And why do gram-negative bacteria lose their crystal violet stain? Because their outer membrane, that is composed of lipopolysaccharide, dissolves during decolorization, allow the dye to escape. Now the slide is immediately rinsed with water to stop further decolorization. The next step is counterstain. Why counterstain is important? To stain the decolorized gram-negative cells pink or red. Secondary stains such as saffronin, diluted carbolfucin is added for 30 seconds to 1 minute. So what is the principle of gram staining? According to pH theory, cytoplasm of gram-positive bacteria is more acidic, hence retained basic dye like crystal violet for longer time. Also iodine form a diiodine complex with primary stain which get retained inside the cell. According to cell wall theory, gram-positive cell wall has thick peptidoglycan layer while gram-negative cell wall has lipopolysaccharide which is more permeable, thus allowing the outflow of crystal violet easily. Alternatively, neutral red may also be used as counterstain, especially for gonococci. The slide is rinsed in tap water, air dried, and then examined under the microscope. What are the modifications of gram staining? Jensen's modification for meningococci and gonococci, Brown and Breen modification for actinomycetes, hot gram staining, cold gram staining, rapid gram staining, fluorescent gram staining, automated gram staining. These are the modifications of gram staining. Why is it important to use fresh cultures for gram staining? Old cultures may not stain accurately due to cell wall degradation. What factors can lead to false gram-negative result? Over decolorization, old cultures, or damaged cell wall. What is gram-variable reaction? When bacteria stain inconsistently as both gram-positive and gram-negative. This is due to various factors like cell wall properties, environmental conditions, or physiological stress. Examples are Microbacterium species, Corynebacterium species, Actinomycetes species, Clostridium species, Nocardia species. How to confirm identity? By special stain like acid fast stain, culture characteristics, biochemical test, or molecular method. Now put a drop of cedarwood oil on the smear and examine the slide under microscope using the oil immersion objectives, that is 100x. So tell me the magnification used to observe gram stain slides. It is 1000x, 10x from eyepiece and 100x from oil immersion objectives. What is the pH range suitable for gram staining? A neutral to slight basic pH is optimal. What happens if the slide is not heat fixed properly? The smears may wash off during staining, but overheating may distort the shape of the cells. What happens if alcohol is not used? Both gram-positive and negative bacteria may retain the primary stain. Why should the slide be rinsed gently with water? To prevent washing away the smear or damaging the cells. What can cause poor staining contrast? Old or improperly prepared stains, thick smears, or insufficient counterstaining. What could cause a mixed gram reaction in a single organism? Cell wall damage, old cultures, or antibiotic affecting the cell wall. Can gram staining be used for all bacteria? No, some bacteria like Mycobacterium species or Mycoplasma species do not gram stain due to their unique cell wall properties. So finally, after gram staining, gram-positive bacteria will appear purple or violet due to primary staining, that is crystal violet, and gram-negative bacteria will appear pink or red due to counterstain, that is saffronin. What is the next step after gram staining in bacterial identification? We have to perform biochemical test, culture or selective media, or molecular diagnosis for further identification. In next videos, we are going to discuss what are the other biochemical tests for gram-positive and gram-negative bacteria and how and what we have to write in our exam papers.